Hi everyone, this is Erin from the DNA Learning Center. I am here to teach you a lab on bacteria and antibiotics. We're in my kitchen today, so welcome. I have a lot of supplies that you might not have at your house, but I'm going to show you how much fun we can have by using some of the lab supplies in a normal environment as well. So right behind me, I have some bacteria, which I grew in tubes. I actually grew these at the lab, and these are two different types of bacteria. Now, I know that the camera's not perfect, but if you're looking at this, do they look the same or do they look different to you if you were just looking at their outward characteristics? To me, they look the same. In fact, what often happens in the lab is that it's possible for the bacteria to get mixed up or mislabeled sometimes. So I'm going to show you a fun experiment where we can identify which bacteria is in which tube using some antibiotics. So have you guys heard of antibiotics before? I'm going to start my presentation to show you some images. So bear with me for a second here. Okay, so hopefully you guys can still see me in that upper corner. And let me show you this picture right here. Okay, so this is a famous scientist, Alexander Fleming. And does anybody know why this scientist is so famous? So Alexander Fleming discovered the first antibiotic, penicillin. Have you guys, I don't know, I can't see the chat right now, but if you have answered what antibiotics are medicines that we can use to inhibit the growth of bacteria or to stop bacterial growth altogether. So I am going to show you right here that bacteria and viruses are often put in the same categories, but they do have differences. Bacteria are living cells. Let me draw this up right here. Hopefully you guys can see this, and maybe oh, there it is. So that's my little bacteria cell. And over here on the side, let me just move this out of the way so I can see. I'm gonna do my virus like that. So viruses and bacteria can have different shapes, and in general, we think of viruses as being much smaller than bacteria. Some bacteria and some viruses have the potential to make humans sick, but not always. In fact, we're going to focus on bacteria today. Most of the bacteria in the world are harmless to humans, although again, there are some that can make us sick. If you have ever had strep throat before, strep throat is caused by bacteria. And I have never had strep throat, but I heard that it's very painful. If you have ever had an infection from bacteria, it's possible if the infection was bad enough that you went to your doctor and they prescribed you a medicine, which we call an antibiotic. If you're sick with a virus though, if there's a virus getting you sick, like the common cold is caused by a virus or the flu is caused by a virus, you cannot use antibiotics to take care of a viral infection. And I'm going to just show you some details about these two to see why we treat them differently. Okay, so both viruses and bacteria have DNA inside. In fact, well, some viruses have something called RNA as well. But let me draw right here. What I'm drawing in the little bacterium there is some DNA. And I'm going to draw some DNA over in the virus too, like that. In addition to DNA, bacteria cells have ribosomes. Do you guys know why ribosomes are so important? Get a few more in here. Oh, didn't mean to do that. So ribosomes are important for reading the information in the DNA and producing proteins. So if you've done any of our classes with us before, You may have seen some images of DNA. Maybe you even did a DNA extraction. But this here, right here, is just a picture of a model of DNA that I have. DNA carries information in it. And so let's see if I can highlight a section of that model for you. So let's say right here, okay? Let's say I'm looking at that section of DNA, and that section of DNA has information, okay, to make what we call a protein. Okay, so this region right here highlighted in red, if that has information to make a protein, I would call that a gene. So that gene carries information. Let's see if I can type the word protein out here. To make a protein. And proteins, the reason we're interested in proteins is because proteins have the ability to impact or influence various traits in cells and within the organism. Okay, so DNA right here is information to produce a protein. Let me draw a little protein there if I can. Let's see if I can make one. So proteins, different proteins have different shapes and they do different things. So that's my little protein right there. So my question is, how do you get from a gene, so the information, to the actual product, to the actual protein? What has to happen in the cell is the cell has to make a copy of that gene into something that we call mRNA, and that mRNA is read by the cell specifically. Let's go back to our bacterial picture. That mRNA is read at that ribosome site. So let's see if I can circle this. I'm going to circle a couple of ribosomes. Oh, I didn't do a very good job. Let's see if I can move that. All right, so those are some ribosomes. What will happen at those ribosomes is the recipe from the DNA will be read and the protein will be produced. So great. All right, now let's take a look over here. Remember our little virus? Okay, we said that viruses do have genetic information. They have DNA. Sometimes they have RNA, which is very similar, and sometimes they have both. But what is the virus missing that even something very simple like the bacteria have? Viruses have no ribosomes, and therefore they're not able to function on their own and produce proteins on their own. We, in fact, don't consider viruses to be living things. They're in this unusual category because they can do some of the things that cells can do, but they can't do everything. So overall, we do not consider them to be alive. They do need to use a host cell in order to do certain functions. Because of the differences between our bacteria and our viruses, if you have a bacterial infection, you're going to respond to that infection differently than if you had a viral infection. One way that we can try to protect ourselves against a viral infection is with something called a vaccine. So a vaccine, you may have had a vaccine before in your life, like if you've gotten the flu vaccine, what you can do is they'll put a little bit of the virus into your body. Often that virus is a weaker form of the original virus or just pieces of it. And what you're trying to do when you're putting that virus in is you're trying to give your immune system some practice on how to fight and combat that virus, so that if your body comes into contact with the fully functioning virus, your immune system has memory, and the hope that is that your immune system would remember how to fight it and could do so and protect you in the future. In our research and in our work with bacteria, let's come back to our original picture over here. Let's see if I can move this. Okay, come back over here. Is that scientists are fascinated with how viruses and how bacteria function. And I mentioned Alexander Fleming to you before because Alexander Fleming is a scientist who was studying bacteria. And he wanted to know how we could help people who had bacterial infections. A lot of times during a war, a soldier might get they might get a wound, and what would happen is that that wound would become infected and the soldier might die of the infection. So during Alexander Fleming's time, this was a major concern. They didn't know how to fight off these infections. And I want to show you what he discovered. Okay. So he's a scientist who I said was working with bacteria, and he had petri dishes. Let me show you a petri dish. Okay, so I've got a petri dish right here. My petri dish has nothing on it. I don't know if you can see that. It just kind of looks yellowish. It just has food for bacteria. He had petri dishes in his lab, and he noticed that on one of the petri dishes, which he had bacteria on, there happened to be a little bit of mold in the region of the petri dish. He noticed that the bacteria on the dish, right around here, do you see where I'm highlighting this area? He noticed that bacteria were unable to grow near that mold. And he had a hypothesis that the mold was producing something that was killing or inhibiting the growth of the bacteria. The mold that he had identified on his plate was a type of penicillium mold. And they ended up naming the substance that this mold produces penicillium. Penicillin. The penicillin was the first antibiotic that anyone ever discovered. Now, when Alexander Fleming was doing his work, it was in fact very difficult for him to isolate that penicillin from the mold. He struggled for a while with that process, and it wasn't until a number of years later that a group of scientists, pictured here, were able to change their method of extraction, and also they ended up using a different type of mold, but still penicillium, and they were able to eventually extract that material and provide it to the general public. So this was an incredible discovery, to be able to find a medicine in a type of mold and then give it to people and save lives. Incredible. Now, what I want you to do today is I want you to use bacteria and I want you to use antibiotics. And I'm going to show you why. Remember how we had those two different types of bacteria? I have this picture for you here. Okay. Okay, so I have two tubes of bacteria, one over here, which the name of the bacteria in this drawing right here, and in one of those tubes behind me, is called MM294. And the name of the bacteria over here is ampicillin-resistant MM294. They look pretty much the same, if you remember, right? They look the same, but genetically, so in their DNA, they are in fact different. This one, the ampicillin-resistant bacteria, has the ability to resist an antibiotic called ampicillin. What does ampicillin sound like? It sounds a lot like penicillin. They're in the same category or group of antibiotics. Ampicillin resistance. I want to explain what that means to you. Think about this. Normally, what do antibiotics do to bacteria cells? Normally, antibiotics inhibit or block the growth or even kill off bacteria cells. So if you're a bacteria cell, what does it mean if you have the ability to resist an antibiotic? If you can resist an antibiotic, it means you can survive in the presence or grow in the presence of that antibiotic. So our goal right now is to see how we can figure out which bacteria is in which tube. I'm also going to use today, let's see if I can move this again, 2 different antibiotics. So I have 2 different types of bacteria and I have 2 different antibiotics. I have ampicillin and I have this other antibiotic called kanamycin. Kanamycin kills bacteria in a different way than ampicillin. Let's come back here for a second. Think about this. If I have ampicillin in my kitchen right now and I have these 2 types of bacteria and I can't figure out which one is which, what would happen if I add ampicillin to this bacteria right here, MM294? What would happen to that bacteria? I would expect that that bacteria would not be able to grow in the ampicillin and would eventually die off. If I add ampicillin to this one over here, the ampicillin-resistant MM294, do you think anything's going to happen? I don't think so. Because these cells are able to resist ampicillin, they should be able to grow just like they regularly do, as if the ampicillin wasn't even around. So really, all we need in this experiment is just the bacteria and the ampicillin. If I add the ampicillin to one of the tubes and the bacteria don't grow, well, I would guess it was just the regular MM294. If I add that ampicillin to the ampicillin-resistant MM294, oh, did I say that backwards? I think I said that backwards. If I add the ampicillin to the MM294, they would be blocked. They wouldn't be able to grow. And if I add the ampicillin to the ampicillin-resistant MM294, they should grow just like they normally do. So why would I even have this other antibiotic in this experiment? Why would I even have that here, this kanamycin? Technically, I don't really need the kanamycin in the experiment, but I really do like to add it in. I want you to think, this bacteria over here, the ampicillin-resistant one, if a bacteria cell is resistant to one type of antibiotic, does that mean that they can resist all types of antibiotics? What do you guys think? No, it does not. So I like using the kanamycin because I'm going to show that just because bacteria can resist one antibiotic does not mean that they can resist all. I'm going to start the experiment. I think it'll make a lot more sense once I start showing you the materials, but I wanted you to have a little bit of background. Let's see, I'm going to stop sharing my screen here. Can you guys see me big again? Okay, let me make that big for myself. All right, so first thing, I'm going to do some measurements. So I have some tips, and I have, this is a pipette. I'm going to be measuring 250 microliters today. And although I do have my bacterial labeled here, I'm just going to mix them up because I want to figure out if we can really use these antibiotics to solve this mystery. So I'll take 2 tubes right here. Just take those. They happen to be the same color. It doesn't matter what color tubes I use. I'm going to label one number 1 and one number 2 so that I can tell the difference. Just going to use a regular permanent marker for this. Okay. I have this little rack over here that I find very helpful, just to hold and organize everything. So let's pick up one of them. I just happen to pick up this. I'm going to remove the cap. I'm going to use my pipette and get a tip. We'll keep everything sterile. I'm going to go to the first stop, gently place this in the bacterial liquid, and release. I'm going to place it in the tube labeled number 1, and I'm going to change that tip to keep everything nice and sterile, nice and clean. Going to get a new tip. Take my second tube of bacteria. Drop the liquid, nice and slow, and add it to the tube. So now my bacteria, I'm ready to roll here. I don't know which one is which, but we're going to solve that using those antibiotics now. I mentioned that we were going to be using Petri dishes. Okay. So these just are Petri dishes with food in them for bacteria, and that's it. I'm going to label my Petri dishes with a permanent marker, and I'm going to draw a line down the center of each dish. And the reason that I'm doing that is ultimately I want to add a little antibiotic, a little ampicillin to one side and a little kanamycin to the other side. So that's just helping me keep organized. I'm going to label one of the dishes number 1, one of the dishes number 2, and I'm going to put an A on the bottom and a K on the bottom. So this is just some prep work that I'm doing to keep everything straightforward. Show you how I labeled them. Just like that. So this is my number 1 plate. This is my number 2 plate. And if you see, they have an A on the bottom and a K. All right. Now it's time to add that bacteria that I have. and just spread it around the surface of the plate. So we get another tip. And I'll pick up my tube. What happened to my tubes? I'll pick up my tube that is labeled number 1. Drop that liquid. And I'm just going to lift. See, I don't want to open the plate too much because I don't want anything in the air contaminating the plate. I'm just going to open the lid a little bit, release the liquid anywhere on the plate, and close. I'm going to do exactly the same thing for the second plate. Taking up my bacteria. Open a little bit and release. All right. I like to use these little sterilized glass beads. Can you guys hear those? We use these a lot in the lab. I'm going to open one of the plates, dump a whole bunch of those beads on there. Get myself a new tube of beads for the second plate. Just like that. Wow, that has a lot of beads in it. Okay. And then I'm going to shake. Normally I would do this for about a minute, but I'll pretend that I did that for a minute so you don't have to watch, okay? Next, I'm going to take those beads out because again, the purpose of the beads was just to move the bacteria across the plate. So each of those plates should now have the bacteria just spread evenly across the surface. Look any different? I'll show you. There's lots of bacteria on there now. If I just let these plates sit on my counter in my kitchen overnight for maybe 1.5 to 2 days, I would see bacteria growing all over the dishes, right? And that wouldn't really help me figure out which one was which. So now I'm going to apply the antibiotic. So there are different ways that you can apply the antibiotic. We happen to buy these little dispensers, okay? I don't know how, there is so much writing on these, they're very hard to actually read, especially from a distance like that. But this one, let me read it for you. This one happens to be kanamycin, and this one happens to be ampicillin. And if you could look inside this little tube, there are so many little pieces of paper stacked on top of each other, and they have all been soaked in the antibiotic. If you've ever used a hole punch and you've seen that little circle fall out after you do it, that's exactly what they look like. I'm going to take these out now and I'm going to put them onto the dishes. So I use a very simple method to do this. I use a little toothpick, okay? What I'm going to do is I'm just going to push out those little pieces of paper and let them fall onto my countertop right now. So this one's the ampicillin. So what I do is I'm just going to let it kind of fall out, and I'll pick it up and show you in a second because I know you're kind of far away from me. This toothpick is not so good. Okay, so I just push out another one. Okay, perfect. Throw that out. So there's nothing dangerous about working with these antibiotics. In fact, if I were to pick them up and touch them, nothing would happen to me, but the antibiotic itself is delicate. If I were to touch the antibiotic with my hands, I suspect that the antibiotic would break down. So I don't want to pick it up because I want it to work in the experiment. And for a long time, we couldn't figure out how to pick up these little, we call them pills, without touching them. And we had, tried to use toothpicks in different ways. And then one of my friends came up with this brilliant idea to use a little bit of glue. So I'm just going to take a glue stick, regular glue stick that you would have in your house. Here's my little new toothpick, nice and clean. And I'm going to get the edge of my toothpick a little bit sticky. I don't need a lot of glue. I just need it to be a little sticky. And I'm going to touch one of those little pills. Let's see if you can see that. I don't know if you're going to, I don't know. It's not going to show you. Okay, but this one's the ampicillin, right? Okay. So I take either of my dishes and I'm just going to open it a little bit. And I don't know if you can see me doing this. I am just gently kind of tapping that little pill or that antibiotic right onto the top of that gel. And look, it sticks. It's kind of stuck on there, which is perfect. I'm going to get a new toothpick. Just use the glue a little bit. Remember to make it a little sticky. And this is my other plate, so I'm just going to put it on the A side, just like I did on that previous plate. Here it is. And now it's time for the kanamycin. So this is my kanamycin one. I'm just going to push them onto the countertop like I did before. 1, 2. Get a little glue. Touch it gently. Okay. Just going to add it just like that. So there's 2 antibiotics on the plate and they're not touching each other. Okay, look at that. Oh, can you see? Let me holding the plate upside down. You guys see? So we have 1 type of bacteria on here, which we're just calling number 1. We have the second type of bacteria on here, which we're calling number 2. And now both plates have a little bit of antibiotic on there. I need to let these plates sit for a little bit because the bacteria need time to grow. I need to have time, so not only can I see the pattern of growth, but I want to also see if those antibiotics are inhibiting that growth. So I'd like to show you some diagrams or some pictures of the expected results. Let's see if I can share my screen again with you guys. Okay, you guys see that? Let's see. I think I've got my Petri dishes here. Okay, yeah, so I didn't label my Petri dishes yet. So what I'll do is I'm gonna make this very simple for myself. I'm gonna draw a line down the center of the dish. If you end up doing this experiment, I just wanna emphasize that the labeling doesn't have to be perfect. I know a lot of students get upset if it doesn't look just right, but it doesn't have to be perfect. We just need a space, an area for each antibiotic. Now remember that well, you know what? Let me write down here like this. If this is MM294, I want to draw the expected results for that bacteria and then versus the ampicillin resistant one. Let me just shrink that up for you. All right. Now we did remember we added the antibiotic. So I'm going to put little circles on there as an example of the antibiotic. Okay, so I got a little antibiotic over here. And what I'll do is just make some copies of that. There we go. So each plate got a little ampicillin and a little kanamycin. And I still need to put my little A and K labels on the bottom, okay? Make sure we know. So we got A, I'll make a second, A, K, K. So we put a little ampicillin on each plate, a little kanamycin. Okay, so let's pretend there was no antibiotic on there. Bacteria would grow all over the plate. But if there's a little antibiotic, what happens is right around the antibiotic, we end up seeing an area of no growth if that antibiotic is inhibiting that growth. So let's think, for MM294, does ampicillin block the growth of MM294? It certainly does. So what you would see is you would see, oh, that's a little too big, hold on. You'd see an area right around the pill, the antibiotic, kind of like that, where in this inner region, the bacteria are not growing. And everywhere else, just make them black, I guess. You would have the growth of the bacteria. That doesn't look very pretty, but you get the idea. Okay, you'd have the bacteria growing all around like that. Okay, I think I have a better idea for the next one. All right, and what about this side? We should get bacterial growth, right? But does kanamycin, what do you think? Does kanamycin kill MM294? It certainly does. So we would have another one of those circles kind of around. You can even see if it lets me erase a little bit. There you go. So these regions where you have no growth that look like these circles, we call those halos. So on the MM294 plate, I would expect to have 2 halos. Now let's consider the ampicillin resistant one. If the bacteria is resistant to ampicillin, would we expect that ampicillin would kill it or inhibit its growth? No. So this is just so cool. The bacteria grows right on top of it. Grows all over the plate, like that. Now, just because bacteria can resist one antibiotic does not mean they can resist another. So the kanamycin should still kill the bacteria. You get that area, that circle, which we call a halo with no growth. And out in this outer region, you get some growth. So that's what I would expect the results to look like. Okay. I did say earlier, I don't know if you heard me, but that ampicillin and kanamycin kill the bacteria in different ways. So the kanamycin, I expect that halo to be a little bit larger as a result. See if I can stop sharing my screen here. Okay, now let me just explain how those antibiotics work before we finish up. What ampicillin does is ampicillin blocks the formation of new cell walls as those bacteria cells are trying to reproduce and divide. Whereas what kanamycin does is kanamycin inhibits or blocks ribosomes from working properly, so cells aren't able to produce proteins. So I hope you guys had fun learning a little bit about bacteria, learning a little bit about antibiotics, and I hope you guys will return and continue to work with us each day. Thank you.